Okay, so let's dive in. We've got this great stack of sources here sent in by you, our listener,
Mm,
all about something really fundamental, but wow, also kind of complex.
yeah.
How do we actually measure the number of microbes in something?
Yeah.
You know, bacteria, yeasts, fungi, the tiny stuff.
Right. The unseeable life that's literally everywhere.
Exactly. Around us, inside us.
And it's a question that pops up constantly, doesn't it? Food safety, environment, medicine, biotech, everywhere.
It really does.
But counting things that small, things that might be dead or just, won't grow where you want them to, well, that's the challenge.
It really is. So our mission today is to unpack these sources, sort of pull out the key idea.
Get a handle on the toolkit scientists use.
Yeah, understand why there are so many different ways to do this, and crucially, what do the numbers actually mean?
Because it's rarely just a simple head count, is it?
Spoiler alert,
Yeah.
definitely not. It's not like counting pebbles in a jar.
Not at all. The complexity comes in like right at the start, deciding what you're actually trying to count. Are they alive? Active?
And then how do you physically detect something you can't properly see?
Exactly.
Okay, so that decision what to count, that brings us to the first, like, major split, right?
The absolute core distinction. You're either going for a total count,
Everything.
pretty much, every
Yeah.
cell, living, dead, active, dormant, the whole lot. Or you're aiming for a viable count.
Which is just the living ones.
Yeah.
Or capable of living.
Yeah, the ones that are alive, metabolically active, or critically capable of growing under the conditions you give them.
And why is that distinction just so vital? Does it really change the approach that much?
Oh, it changes everything. Think about checking drinking water versus checking yeast in bread dough.
Okay.
In water, maybe you want a total count. Just get a baseline microbial load. Even dead cells might indicate past contamination.
Right, overall quality.
But in dough,
Right.
you only care about the viable yeast, the ones making the gas, making it rise.
Dead yeast cells are useless there.
Exactly. Counting them tells you zip about your bread. So the purpose dictates the approach.
Okay, that makes total sense. Let's dig into the total count methods first then. Trying to measure everything. The most obvious way seems, well, just looking at them.
That's the starting point. Direct microscopic counts. The classic method. Put a sample under the scope, count what you see in a defined area.
But how do you manage that? They're tiny and sometimes moving,
Good point. So you use special slides, counting chambers.
like for blood cells, hymocytometers.
Those, yeah, or often shallower ones for bacteria, like the Petroff-Hauser chamber. They have these tiny etched grids on them.
A grid.
A very precise grid, covers a known area, holds a known volume. You put your sample on, count the cells in a few squares, do some math, get cells per mil.
Okay.
Or you can stain them first, make them easier to see. There are stains like Newman's stain used for milk, or you just add stain to the chamber.
So you're literally counting in a tiny, tiny box. What's good about that?
Well, it's fast. You can get an answer pretty quickly, minutes maybe.
Okay.
And you can actually see the cells, their shape, their size. That can be useful. Plus, the equipment needed is pretty basic. Microscope, slide.
Sounds straightforward. But the downsides, there must be some.
Oh, plenty. Getting an even spread on that slide?
Yeah.
Tricky. You have to count lots of squares and average it, which gets tedious fast.
I bet.
Debris in the sample. Looks a lot like cells sometimes. Very small bacteria are hard to spot, especially unstained.
And the moving ones?
Yeah, motile bacteria are a nightmare. You often have to add chemicals like formaldehyde or methylene blue just to stop them swimming around so you can count them. It's labor intensive if you have lots of samples.
Okay, so manual counting is slow and fiddly for big jobs. Are there automated ways, faster ways to count everything?
Absolutely. That's where particle counters come into play. Automating the process.
Like the Coulter counter. I feel like I've heard of that.
Yes, the Coulter counter is a classic. The principle is neat. You suspend your cells in a conductive liquid.
Salt water, something like that.
Usually, yeah. Then you draw this liquid through a tiny hole, an aperture. There are electrodes on either side.
Okay.
As a cell passes through that hole, it disrupts the electrical current slightly because the cell isn't as conductive as the liquid.
Creates a blip.
Exactly. A tiny voltage pulse. And the size of that pulse is related to the size of the cell. The machine counts the pulses.
Clever. And it can tell size, too.
It can give you a size distribution, which is pretty useful. And it's fast. Thousands of particles per second. Very reproducible.
Sounds great. Limitations.
Well, it works best for larger things. Yeasts, protozoa, bigger bacteria, maybe many bacteria are right on the edge of its detection limit.
Yeah.
And the big one, It counts any particle of the right size. It can't easily tell a bacterial cell from, say, a tiny bit of plastic or dust if it's the same size.
So not great for dirty samples.
Not without pre-treatment, no. And yeah, they're expensive machines.
Okay.
Yeah.
What about flow cytometry? That sounds even more high-tech.
Flow cytometers are definitely sophisticated. Again, you suspend cells in liquid, but here they flow single file, like really fast, through a narrow tube.
One by one.
Exactly. Past a laser beam. As each cell crosses the laser, it scatters light. Detectors pick that up.
Just scattering.
Well, that's the basic count. But if the cells are naturally fluorescent, or if you've stained them with fluorescent dyes,
They glow.
they glow when the laser hits them. Different detectors measure that fluorescence.
So you could potentially count different types of cells if you stain them differently.
Precisely. Modern machines use multiple lasers, multiple detectors. You can count different populations, maybe even look at cell health with certain dyes. Some can even physically sort the cells.
Sort them. Wow. Advantages.
Speed. Incredibly fast. Thousands of cells per second. High sensitivity, high specificity if you use the right stains. And that sorting capability is unique.
Disadvantages, besides the cost, which I'm guessing is high.
Very high cost, yes. Complex instruments, too, need skilled operators. And sample prep can be a bottleneck. Getting everything into a single cell suspension, staining, that can take time.
Right. Okay, one more in this sort of automated category. Image analyzers.
Think microscope plus camera plus smart software.
Okay.
You prepare a slide, the system takes pictures, and the software analyzes the images. It identifies and counts particles based on criteria you set: size, shape, maybe color if stained.
So automation for microscopy.
Kind of. You get some visual info, but computation does the counting. The potential plus is selectivity. You might tell it only count things this big and ignore smaller stuff.
But the downsides.
Still have the microscopy issues, like getting an even spread. The software can be pricey. And analyzing lots of images for many samples can still take a fair bit of time.
Okay, so those are direct counts looking or detecting individual particles. But sometimes that's just not practical. What then?
Then you move to indirect total counts, measuring something else that relates to the total number or mass of cells.
A proxy measure.
Exactly. And probably the most common one here is measuring turbidity.
Just how cloudy the liquid is.
Basically, yeah. You use a spectrophotometer, shine light through the sample, measure how much light gets through that's transmittance, or how much is blocked or scattered that's absorbance or optical density, OD.
And more cells means more cloudy, higher OD.
Generally, yes. As cell concentration increases, they scatter more light, so the OD goes up.
Simple enough.
But there's a catch. Unlike dissolved chemicals that absorb light following Beer's law nicely, cells mostly scatter light.
Okay.
And because it's scattering, the relationship between OD and cell number isn't perfectly linear, especially at high concentrations.
Why that?
At high densities, light scattered by one cell might hit another cell before reaching the detector, or cells might shadow each other. So the OD reading doesn't increase as much as you'd expect. It starts to curve off, usually above an OD of maybe 1.0 or so.
Oh, so not a straight line forever. That's important.
Very important to remember. You can measure this on different scales. Absorbance, percent transmittance, percent per C, sometimes Collet units. You can convert between A and per C, but consistency is key.
Does the color of light you use matter? Like red light versus blue light?
It does. Shorter wavelengths, blue light gets scattered more effectively, so it's more sensitive for low cell numbers.
It's your sensitivity.
But that nonlinear problem, it starts happening sooner at lower concentrations. Longer wavelengths, red light, are less sensitive overall, but they often give you a more linear relationship over a wider range of cell densities. That's often better if you're tracking a whole growth curve.
Okay, so OD gives you a number, but it's not directly cells per mil.
Yeah.
How do you connect the two?
Standard curves. Absolutely essential. Non-negotiable.
Right. Explain that.
You have to prepare samples of your microbe at different known densities. For each one, you measure the OD and you determine the actual cell count. Or, more often, the dry cell mass using another method.
Like microscopy or weighing them?
Exactly. Then you plot your OD readings against those known counts or masses. That graph is your standard curve.
And then you use that graph for unknown samples.
Yep. Measure the OD of your unknown, find that value on the graph, and read across to estimate the cell count or mass.
And I guess that curve is pretty specific.
Extremely specific. It only works for the exact same microbe grown in the exact same medium under the exact same conditions measured at the exact same wavelength on the exact same instrument. Change anything, the curve changes.
Wow. Lots of variables.
And OD generally correlates better with total cell mass than with the precise cell number because cell size can vary.
You mentioned nephelometry briefly, too. Different from OD.
Nephelometry also measures scattered light, but instead of looking at light loss from the main beam, it measures light scattered off at an angle, usually 90 degrees.
Why do that?
It tends to be more sensitive, especially for very dilute suspensions where OD struggles. And importantly, it's not really affected by the color of the growth medium.
Ah, that's useful.
Yeah, laser-based nephelometry in microplates is becoming quite popular for sensitive growth monitoring.
Okay, so turbidity, quick, easy, non-destructive sometimes, but struggles with colored or already cloudy samples and that linearity issue.
That's a good summary. And cell clumps really throw it off, too.
Right, so what if your sample is really turbid or full of particles? OD is out. Other indirect ways,
You can measure stuff inside the cells, assays for cellular components.
like protein or DNA.
Exactly. Total nitrogen, total protein, nucleic acids.
Yeah.
Those are common. The idea is if you know roughly how much protein, say, is in an average cell or per gram of cells, you measure the total protein in your sample and estimate the biomass from that.
And you do that.
Usually collect the cells, wash them to get rid of medium interference, break them open, do a chemical assay for the component, often read on a spectrophotometer again.
Can you measure things specific to certain groups?
Yes. Like
Yeah.
chitin for fungi, though some other things have chitin too. Or ergosterol, that's a big one.
Ergosterol, that's specific to fungi, right?
Right.
Our sources mentioned it for soil.
Perfect example. Fungi in soil are mostly thread-like hyphae, plus spores. A plate count doesn't work well for biomass, as we'll discuss, but ergosterol is in the fungal cell membrane. Measure the total ergosterol in a soil sample, and you get a really good estimate of the total mass of fungal material there, much better than counting colonies.
That makes sense. It directly relates to the amount of fungus, so pros and cons for measuring components.
Pro. Works when OD fails, in turbid or particulate samples. Cons. Generally more work, more time than OD. Assays can be fiddly, subject to interference. Non-microbial sources of the component can be a problem like insect chitin if you're measuring fungal chitin.
And the amount in each cell isn't always constant.
Right, it can vary with growth stage conditions. So again, you absolutely need a standard curve relating your component measurement back to actual biomass or cell number determined under your specific conditions.
Lots of calibration needed. Any simpler physical methods?
Well, there's the sedimentation tube method. Spin down your sample hard in the centrifuge tube, measure the volume of the packed cell pellet.
Just measure the height of the sludge at the bottom.
Pretty much. It's quick, easy, low-tech, but not very accurate or sensitive. Useless if your medium has particles that also sediment.
Right.
Sources give a rough guide. Maybe 5 cubic centimeters of wet pellet is about 1 gram dry mass, assuming 80% water. And you need to spin harder for bacteria than for yeast or mold.
Okay. And the most sort of intuitive physical way, just weigh them.
The dry biomass assay. Very fundamental, especially in things like fermentation studies. You take a known volume, collect the cells.
Centrifuge or filter.
Yep. Then wash them
Why?
to get rid of salts or sugars from the medium that would add artificial weight. But don't overwash, or you might lose stuff from the cells.
Checking balance, then weigh.
Then dry them completely, usually in an oven at a 1 or 5 Celsius until the weight is constant. That can take 12, 24 hours. Then weigh the drag mass.
Filtration seems easier in collecting than spinning sometimes.
For yeast or molds, maybe, but bacteria, tiny. They clog filters really easily. It can be a nightmare. And you need filters that don't lose weight themselves during drying, like glass microfiber ones.
Okay, pros and cons for dry weight.
Pro. Gives you a direct measure of dry mass, a key parameter. Conceptually simple. Cons. Slow results because of drying, not very sensitive for low concentrations. Useless if the medium has particles.
And that tip about standard curves.
Yeah, good point from the sources. If you're making a standard curve relating OD to dry mass, it's often better to measure the dry mass accurately for your most concentrated sample, then calculate the expected mass for the dilutions based on the dilution factor. Trying to weigh tiny amounts from dilute samples is often inaccurate.
Makes sense. Okay, last one for total counts. Relating metabolic activity like products made or nutrients used.
Less common as a general total biomass method, but used as specific research. If you know the metabolism well, you might relate, say, CO2 production rate to the amount of active biomass using a yield coefficient.
But that relies on the microbes behaving predictably.
Heavily. The yield coefficient needs to be stable, the assay's accurate. If metabolism shifts, it gets unreliable fast.
Okay, that's a lot on counting everything. Let's shift gears. Viable counts. Now, we only want the living active ones. That feels different.
It is. Now, the method needs to tell you something about the cell's state. Can it divide? Is it respiring? Membrane intact?
First one listed, most probable number, MPN.
MPN is statistical, not direct counting. You do serial dilutions of your sample.
Okay.
But then for each dilution, you inoculate multiple tubes of liquid growth medium, say 3 or 5 tubes per dilution.
Multiple tubes.
Yes. After incubation, you just look at how many tubes at each dilution show growth, turbidity, gas, whatever.
So you don't count colonies?
No, just positive tubes. Based on the pattern of positive tubes across the dilutions, like all 5 positive at 10:1, 2 positive at 10:2, 0 positive at 10:3, you look up the most probable number in a statistical table.
A statistical estimate. When would you use this?
It's good for very low concentrations, like water testing, or for microbes that just don't grow nicely on solid agar, some algae, certain bacteria.
Advantages?
Good for low numbers, conceptually simple.
Disadvantages?
Low accuracy, it's an estimate, takes time, 24 hours plus for incubation, needs lots of tubes, media, incubator space, labor intensive. The classic fecal coliform test in water is often an MPN.
Sticking with low concentrations, especially water. Membrane filtration.
Right. Very useful for things like drinking water. You filter a known, often large volume of water through a sterile membrane filter. The filter has tiny pores that trap the bacteria.
Catches them on the surface.
Exactly. Then you take that filter paper and lay it right onto the surface of an agar plate with growth medium.
So the filter acts like the plate surface.
Kind of, yeah. You incubate it, and each viable cell trapped on the filter grows into a visible colony. You count the colonies.
So you concentrate the cells first.
Precisely. Makes it great for low numbers. Accurate, relatively simple, uses less glassware than MPN. You can use selective media on the plate too. Downsides.
Takes time for incubation, 24 hours plus. And the filter holder needs careful sterilization between samples, which slows things down if you have many different samples.
Okay, and now the one everyone thinks of. Dilution plate methods. Pour plates, spread plates.
The absolute classic.
Yeah.
Serial, dilute your sample in a suitable liquid that keeps the cells alive but doesn't let them grow.
Sterile saline or buffer.
Usually. Then take a small, known volume of a dilution, put it on or in an agar plate, sped plate, pipette onto solid agar, spread it evenly. Pour plate, mix with melted, cooled agar, pour into the dish, let it set.
Then incubate.
Incubate. And each viable cell that can grow on that medium under those conditions forms a visible colony forming unit, a CFU.
Little clump of cells.
Yeah. You count the CFUs, factor in the dilution and volume plated, calculate back to the original concentration.
Seems simple. Count the dots, do the math.
But the catches, I know there are big ones.
Huge ones. First, clumps. If 2 or more cells stick together, they only form one colony, so you underestimate. Second, and this is probably the biggest limitation of plating, no single medium grows everything. Absolutely critical point.
This is that statistic from the sources about environmental samples.
Exactly. This is massive. Estimates suggest that for many environments like soil or water, maybe only 1% or even less of the total cells you see under a microscope will actually grow on a standard lab plate.
1%.
Or less. Many microbes have really specific needs we don't meet. Others might be stressed, dormant, alive, but just not ready to grow under those artificial conditions. This leads to the idea of viable but non-culturable cells, VBNCs.
Alive but won't grow on the plate.
Exactly. They're a huge blind spot for plate counts. So a plate count tells you the number of culturable organisms. It's almost always a significant underestimate of the truly viable population and a massive underestimate of the total population.
So CFU count is really culturable units, not necessarily viable cells.
That's a much better way to think about it. And you aim for plates with 250 colonies, ideally. Too few isn't statistically sound, too many, and they just merge together, too numerous to count. TNTC.
Okay, so plating has major limits on telling us who's really alive. What about methods that assess viability without needing colony growth?
Now we're talking things like the ATP bioluminescence assay.
ATP, the energy molecule.
Exactly. ATP is the main energy currency in active living cells. Dead cells lose their ATP pretty fast.
Oh, you measure it.
With the firefly enzyme, luciferase. You add luciferase and its substrate, luciferin, to your sample extract. If ATP is present, the enzyme uses it to make light.
More light, more ATPs, more active cells.
That's the principle. It's great because it works in murky or colored samples where OD fails. It's super fast results in minutes.
Right.
And Very sensitive.
Sensitive.
Yeah, can detect tiny amounts of ATP. You can even filter a sample to concentrate cells, then measure ATP, making it even more sensitive. And there are tricks to measure bacterial ATP specifically by getting rid of ATP from other cells first.
Sounds powerful. What are the challenges?
Well, it's a precise technique, needs careful handling. The main biological issue is that the amount of ATP per cell isn't constant. It varies hugely between species, with cell size, growth phase, stress levels.
Ah.
So it's a good indicator of total metabolic activity or active biomass, but converting that light reading into an exact cell number is difficult and often unreliable, and the equipment's fairly expensive.
Okay. We mentioned microscopy with stains for yeast viability earlier. Methylene blue, are there others?
Yes, viability or vital stains used with microscopy. These dyes react differently with living versus dead cells, usually based on membrane integrity or enzyme activity.
Examples.
Fluorescent stains are powerful here. You need a fluorescence microscope. A common combo is carboxyfluorescein diacetate, or CFDA. Living cells take it up, enzymes inside convert it to a green fluorescent molecule that gets trapped inside.
So living cells glow green.
Right. You combine that with something like propydium iodide, PI. PI is red, and it can only get into cells with damaged membranes, dead or dying cells where it stains the DNA. So you see green viable cells and red dead cells.
Green means go, red means stop.
Got it.
But didn't you say some common fluorescent stains are not good viability indicators? Like DAPI?
Absolutely crucial point and a common mistake. Stains like DAPI and acridine orange are used all the time for total bacterial counts, especially in environmental samples. They bind to DNA and RNA for acridine orange and make cells glow brightly, making them easy to see.
Kaput.
But they bind nucleic acids whether the cell is alive or dead. Acridine orange might give a hint of activity sometimes by staining RNA differently, but DAPI does not indicate viability or metabolic activity. You could be counting DNA in cells that died long ago, or even just bits of free DNA stuck to particles. The sources warn that maybe only 2% of DAPI-stained dots in some samples are actually live cells.
Wow. So a bright spot doesn't mean a living microbe necessarily.
Not with those stains, no. For better microscopic indicators of activity, people use things like CTC, a dye that turns fluorescent red when it's reduced by active respiration, or fluorescent probes targeting 16S ribosomal RNA. Active cells making lots of protein have lots of ribosomes, so high RNA is a better sign of life than just DNA.
What are the downsides of these staining methods with microscopy?
Generally labor-intensive, counting fields manually, hard to do many samples, can be prone to interference from background fluorescence or particles in the sample.
Okay, shifting to the molecular level. PCR-based technologies, how do they fit in?
PCR, polymerase chain reaction, is mainly about detection and identification, not usually precise counting, although quantitative versions exist. Its strength is finding specific DNA sequences.
Like a genetic fingerprint.
Exactly. You amplify a target sequence unique to a certain microbe or group, design primers, cycle the temperature, polymerase enzyme makes copies. Billions of copies from just a few starting molecules. Makes it super sensitive for detecting even rare organisms like pathogens. You typically see the result on a gel.
And real-time PCR.
qPCR.
That's the quantitative version. Real-time PCR, qPCR, uses fluorescent dyes or probes that glow when the target DNA is amplified. The machine tracks the fluorescence increase cycle by cycle. How quickly the signal appears tells you how much target DNA you started with.
Also gives you a number.
It gives you a quantity of target DNA, yes. It's much faster, often under an hour, no gels needed. Multiplex PCR lets you look for several different targets at once.
Sounds amazing. Sensitive, specific, fast.
It is all those things. Huge advantages.
But the big catch for viable counts.
The Achilles heel. Standard PCR detects DNA, not living cells. DNA can hang around long after a cell dies, so you can get false positives if you're asking, is this dangerous bacteria alive here right now? You might just be picking up remnant DNA.
So not a reliable viability test on its own?
Not usually, no. Plus risks of lab contamination giving false positives, or inhibitors in the sample giving false negatives. Quantification can be tricky to get really accurate. Needs trained people, expensive gear.
The sources mention that HANA device. Yeah, the HANA Way handheld advanced nucleic acid analyzer. It's a neat example of portable PCR. Small, battery-powered, designed for field use like rapid biothreat detection. Shows the tech is getting more accessible, faster results, under 30 mins.
Okay. And finally, biosensors.
That's a broader term. Instruments designed to measure microbial activity rather than count cells directly. Might measure a chemical change caused by a microbial metabolism, like a change in electrical current. The weight of change relates to the amount of active biomass, often quite specialized.
Ugh, okay. So we've gone from microscope dots to light scattering chemistry, ATP, DNA.
Yeah.
It's definitely clear there's no single best method.
Absolutely not. That's the core message from these sources. The method depends entirely on the question and the sample.
So what are the key things someone needs to think about when choosing?
Number 1, purpose. Total count, viable count, active count. Need high sensitivity for low numbers.
Okay, what's the goal then?
Practicability. What's your sample like? Is it clear or cloudy? Colored. Got particles. How does the microbe grow? Single cells, clumps, filaments, spores. What kind of microbe is it? Easy to culture. How big?
Sample characteristics.
Resources. What equipment do you actually have? How much time? How much labor? Budget.
Makes sense. Let's use the source examples. Fungal biomass in soil. Why is plating bad?
Because soil fungi are mostly these long threads, hyphae plus spores. One colony on a plate could come from one spore, or a tiny piece of hyphae with hundreds of cells. You can't tell. The source says a plate count is basically just a spore count. Tells you nothing about the actual mass of fungal threads.
Right, so instead you measure...
Ergosterol, that fungal membrane chemical. Total ergosterol in the soil gives a much better estimate of total fungal mass.
Okay, what about that tricky growth curve example? Dark medium, lots of particles. OD is out.
Totally out. Can't use turbidity. So if you need total biomass, you'd likely switch to dry biomass, collect, wash, dry, weigh.
Makes sense. But then they added another complication. The particles are substrate that the microbe eats.
Ah, yes. That's a great illustration. Now, dry mass is tricky too because the mass of the medium particles is changing as they get consumed. You can't just subtract a fixed blank weight.
So what do you do?
In that really specific, tough case, dark, particulate, medium, consumable particles, the sources suggest measuring total protein might be the best bet. You measure total protein over time, relate it back to biomass using a standard curve.
Oh, you might have to make separately.
Yeah, maybe grow the microbe in a similar clear medium just to establish that protein to biomass ratio accurately. Shows the kind of workarounds needed sometimes.
Definitely highlights the ingenuity involved. So bringing this back to you, the listener, getting a handle on all these methods really helps you interpret data you see, right?
Absolutely. You understand the effort behind the numbers. You see why different studies might get different results for seemingly the same thing. One did a plate count, another a total microscopic count. You appreciate the limitations.
It's clear that measuring microbes is incredibly varied, requires this huge toolkit, and every single tool has its strengths, but also significant weaknesses.
Exactly. No magic bullet. It's always a trade-off based on the question, the bug, the sample, and the resources.
And when you put all those challenges together, the counting difficulties, the culture bias, those viable but non-culturable cells, DNA methods detecting the dead.
It really makes you wonder, doesn't it?
What does any single microbial number we report, a CFU count, a total cell count, what does it really represent? And considering everything we might be missing with any one method, how much of the microbial world, its true scale, its diversity, its activity, are we still fundamentally underestimating or just misunderstanding?
That is the big question, isn't it? Something to think about.